Good evening, ladies and gentlemen. Welcome to the 14th CeMM Landsteiner Lecture. We are very happy that you joined us for this semi-virtual event. I have the pleasure in this room to have listened to live music since a long time. So we had a duo number one by Ludwig van Beethoven, and the musicians were Anna Lang on cello and Doris Freimüller Auinger, and I would like to thank them very much. We'll hear more later. This whole lecture should have happened a year ago, and then COVID happened, and this year we decided to have this hybrid mode. In this hybrid mode, the people in the room, there are a few people in the room, all are tested heavily and vaccinated. And so we chose Beethoven because last year, as you know, was the 250th anniversary of Beethoven's birth. And Beethoven came to Vienna, like many other researchers that are online today, and are participating to the discoveries and the research that are happening in Vienna and other places in the world. He came to Vienna as a young man looking for his fortune, and as you know, he has changed the perception of what young foreigners can do for this country, being the first one perhaps who really established a very mature eye-to-eye relationship with the people in charge. And so for us, Beethoven has a special role, and I'm very thankful for the musicians for sharing their art with us tonight. Very briefly, I'll tell you a few words about the Center for Molecular Medicine. We belong to the Academy of Sciences. We are situated in the center of a very large research hospital, a first-rate place for medically oriented research. We are about 200 people distributed through different parts. In the building, there are 140 people, and many are members of CeMM, but in adjacent adjunct laboratories. So the purpose of CeMM is to make innovation in molecular medicine, and it's a fairly young place, a little bit older than the age of Beethoven when he came. They are, because I'm the oldest in the team, 33 years of age in average, and we cover almost 50 nations. So we are proud of being able to cope with a budget that is half brought by the federal ministry through the Academy of Sciences and half through grant writing and third-party funds. And clearly, we have been very happy to have 12 ERC grants that we take as a sign that we're doing something right, as well as a few additional important international grants that put us on the map. Now, what are we doing in concrete? We try to do as much innovation as possible, meaning that we don't do derivative research. So we are trying to do free-minded basic research. But we do this in the medical context. So the entire campus speaks of medicine. There is a lot of unmet medical need around us. And that inspires, if you want, the spark of ideas that people have here. And I think this relationship between being free to do very new kind of stuff, but being embedded into a very important and heavily researching surrounding is a recipe for being able to achieve a lot of new sort of results and initiatives in molecular medicine. Very important is that we work on post-genomic technologies, meaning that we are taking the human genome for granted. We start from there, and we have a few diseases for which we are focusing in particular, and such as immune disorders, cancer, but also metabolic disorders and infections. A very important part of our mission is to train young scientists at all levels. When PIs start, they're usually very early in their career, but also, of course, postdocs and students. And we are overall happy to have contributed or to contribute to what we call the 5P medicine, and that is a medicine that is both precise, personalized. You hear this is now very important, becoming more and more important, but also predictive and preventive, so that in future medicine will be quite different than what we have seen so far. So in one of the things that we're doing is we are trying to foster young investigators. And I would like to start by introducing to you the Denise Barlow Award. Behind me, you see a picture of Denise Patricia Barlow. Denise has been a principal investigator at CeMM for 10 years, a spectacular scientist doing very, very fundamental work on genomic imprinting, in particular, understanding how long coding RNA participate to genomic imprinting. And Denise has been a pioneer. She was the first female principal investigator at the IMP at the time that the IMP was built, IMP, Institute for Molecular Pathology in Vienna in the 80s. And then she also was a member of the Max Perutz laboratories, and she was also associated with IMBA for some time. So she was in 4 of the biomedical institutions in Vienna. And when she sadly and prematurely passed away 2 years ago, we decided to make an award for the kind of people that she cared most for, which are young investigators. And in particular, we thought to invent an award that would help young investigators do the transition from the PhD to the postdoc. As you know, each transition brings risks that people change career, do other things, but we really think that it is important to foster young people to do their postdocs, and hence this award. And the award is given to anybody, not anybody, to the best person, to the person that has done the most interesting PhD globally, the PhD work in one of these 4 institutions. And there is a committee of 8 individuals that are helping with this decision, 4 directors, and then very prominent RNA biologists and genomic, epigenomic experts. And I'm very, very happy to use, if you want, the Landsteiner Lecture as a platform to present to you the winners of the Denise Barlow Award, the 2019 winners, so the one of last year, Matthias Muhar, previously with Hannes Zuber at the IMP, now in Zurich, and he will tell us a little bit about what he's doing. But also Julia Batki, also from the same year, from Julius Brennecke lab at IMBA, another fantastic researcher. And then this year's award-winning PhD student, Annette Romanowska, who is with Alvin Köhler at the Max Perutz Laboratories. And we have the pleasure of having Matthias here with us. So I would like to start with Matthias. Matthias, welcome. Congratulations from the entire community. We are very happy that you accepted the award, and we hope it will help you for your future career. You want to say something?
Yes, of course. First of all, I want to say, of course, thank you, Giulio, and a big thanks to everyone involved in organizing this event and the prize. It's really a tremendous honor to be here. And of course, I also have to thank the many people involved in my previous work, especially my mentor, Johannes Zuber, and Anja Ebert, and many other collaborators. So in this work, we actually looked at a phenomenon that is also very close to what Denise Barlow was researching, and that is how genes' activity is regulated at the level of transcription. And we particularly looked at the level, at this phenomenon in cancer cells, and wanted to dissect the mechanisms, how different programs go wrong in cancer cells.
Thank you very much. Good luck with your future work. We have connected with us online Julia Batki. Hello, Julia. Congratulations for this award. Where are you, and would you like to say a few words?
Thanks, Julia. I'm currently in Berlin, where I'm a postdoctoral fellow at the Max Planck Institute for Molecular Genetics. So first of all, I would like to express how honored I am to receive the Barlow Award, especially that I know what an inspiring scientist Denise was. I was fortunate enough to meet her at the Vienna RNA retreat. And in my PhD work, I identified and characterized a novel protein complex, which we named Swings. And this complex, I found that it's essential for silencing transposable elements, or so-called jumping genes, in the fruit fly. And this work was not possible without all the tremendous support and guidance I received from my PhD mentor, Julius. And I'm also grateful for all the collaborators and the Brennecke Lab, the entire Brennecke Lab, for their essential contribution. And thank you very much for the committee.
Fantastic, Julia. Congratulations. May the award help you. We are all fevering for your future research. Thank you for sharing your memories. And now I would like to move on to this year awardee, Annette Romanowska. Annette, where are you? And say a few words, please.
Currently, I'm back home in my home country, Latvia. And first of all, I want to say that I'm really honored and really happy to receive this award. And I want to thank the jury and the organizers. But the biggest thanks, of course, goes to my supervisor, Alvin Kohler, because without him, this would not be even possible. And together with Alvin, we studied the inner nuclear membrane, the membrane that encloses the genome. And previously, it was assumed that this membrane is metabolically silent. But we found that the inner nuclear membrane is able of active lipid metabolism and can even generate lipid droplets inside the nucleus.
Fantastic. I can tell you that the jury had difficulties in finding the only winner, because you all have done fantastic work, but this particular work on the nuclear membrane and lipid organization was spectacular. So thank you very much. Also from us and from the community, good luck with your future endeavors, and may you also become one of these important let's say leaders in the community, because we need people like you. So thank you to all three awardees. I will now take one of exemplary containers. Inside you have the check for 5,000 euro, as well as the Urkunde, the diploma that goes with it. I symbolically give it to Matthias. Imagine it will be in your hands. You will receive it per super post in due time. So Matthias, Annette, Julia, congratulations from all of us. May that bring you good luck and viel Erfolg for the future.
Thanks.
Bye-bye. So I clap the hand for everybody here. Thank you very much. Now we move on to the second part, and this is introduced by 3 écossaises of Beethoven, and join me in listening to the music. Fantastic. Very, very nice and entertaining and funny, these 3 écossaises, especially the one with the bass clarinet we loved. And so I would like to thank Anna Lang and Doris Freimüller-Auinger for being with us tonight and refreshing our memories of how nice it is to listen to good live music. So thank you very much. And now we move to the core part of the evening, the 14th Landsteiner Lecture. It's a very, very big pleasure to be able to introduce Sarah Teichmann to you. Sarah Teichmann is something like a legend in our field, as she has been a pioneer of understanding protein function, and throughout her incredibly stellar career, she has not missed a beat in contributing very, very important chapters in the increasingly complex understanding of organization from the gene sequence to the cell, the body, and the organs. Sarah went to the European school in Karlsruhe, itself an amazing high school. I know this because several people from the European Molecular Biology Laboratory were driving an hour every day to have the children go into that school, and she graduated, of course, with the best possible grades. She then moved to Cambridge, studied biochemistry at Trinity College, where she again graduated with amazing honors. And then she joined the laboratory of Cyrus Kothia, one of those persons that have put order in our understanding of proteins. I remind you that proteins are what most genes are doing. And so they are, if you want, the level of organization that comes in translating the genomic information into 3 dimensions. And Cyrus was one of those persons that have really tried to catalog the type of folds and how they would be arranged, but doing also from an evolutionary point of view. And this is something that Sarah, I think, treasured because, as Dobzhansky, a famous geneticist, has said in the past, nothing in biology makes sense if not in the light of evolution. And so understanding where the genes come from, their duplication, and how that results in the multiplicity of protein is something that Sarah studied with Cyrus. She then moved on to another incredibly important person in biochemistry and computational biochemistry, Janet Thornton. Janet, a person that, if you want, on top of what Cyrus Kothia has done, was classifying enzymes, and classifying enzymes by their 3D structure, but also by the way that they arrange the center core of the enzyme. And Sarah here had also very important contribution. And so she was trained by 2 of the towering figures of, if you want, modern global biochemistry, a biochemistry that doesn't only look at the individual case, but is able to look at the bigger picture. And then with only 26 years of age, she became a principal investigator, and she's been in Cambridge since then. Now she is the head of Cellular Genetics at the Sanger Wellcome Center, where she's had an incredibly vigorous research program. And I can tell you this about Sarah. She took where she had already arrived with Janet into a yet a higher level of organization by looking at protein complexes. And I had myself a very strong and have a strong interest in protein complexes. And I can tell you that Sarah is the individual who put order into the logic of these complexes. We were always looking at some general principles, but she actually did it in a very systematic way, to the point that one of her key papers was called the periodic system of protein complexes. From then on, the step in trying to use this knowledge of how 3-dimensional arrangements are, if you want, mirrored in the genome, led her to understand genomics better and better. What are the repertoire of genes expressed in the individual cells, and what does that mean? And so she's been pioneering, if you want, the science of understanding what gene expression actually means by taking always bird's eye views and understanding principles that other people had not seen, and has been, in the last years, a pioneer in what we call a single cell analysis as an instrument to understand what the repertoire of genes expressed in a single cell tell us about the identity of the cell and the identity, of course, of the organ and the purpose of the organ and the person of the organ within the organism. So to me, Sarah represents a person that puts order into biology, is one of those people that are not just making contributions that pass in time and that are little advancement. She's really one of those persons that explain to us how biological matter is organized. So a fantastic achievement. Dear Sarah, we are very, very happy that you accepted this invitation. We are sorry not to be able to house you in the beautiful Austrian Academy of Sciences, the Festive Hall, one of the nicest rooms that already Beethoven has played several times. I promise that we will invite you back in order to play that room, maybe again with the musicians of tonight, to enjoy what Vienna offers with a combination of science, art, into a beautiful, and music into a beautiful setting. So thank you, and we are looking forward to the 14th lecture.
Giulio, thank you so much for that kind introduction and for the very generous welcome here in Vienna, so to speak. It's a tremendous pleasure and an honor to be giving this Landsteiner Lecture in Vienna, in your institute and in your city. What I'll be talking about today is mapping the human body one cell at a time. And that's what my research is about in this period. Our body consists of organs and systems, and within each organ, there are different tissues. And each of those consists of a fantastic diversity of cell types. Each cell has pretty much the same DNA content. And yet some cells turn into fibroblasts, which you can see on the left-hand side. Other cells turn into neurons. And what is it that makes each cell look so different and adopt a different function? It's an invisible machinery that interprets our genetic code and transcribes the genes into RNA and the RNA into protein. And it's the resolution revolution in genomics that's been happening over the past dozen years or so that allows us to resolve the complete complement of nucleic acid, DNA, the epigenome, RNA in each single cell. Now, why is that important? It's because historically, we've looked at cells mainly using microscopes. And the microscopes have shown us the morphology and shape of cells and given us information about a few key molecules in each cell. With the resolution revolution in genomics, we can see the entire complement of the message, the RNA messaging molecules inside each single cell, which basically allows us to resolve the function better than a microscope in an unbiased way, in a very broad way, because what we can do is take a tissue sample, break it up into individual cells, make a cell suspension, and then isolate the RNA inside that cell, read it out with next generation sequencing, and interpret the data with computational methods, artificial intelligence, bioinformatics. And what that tells us about each cell is an incredibly rich information about the molecules that are active in that cell, which gives us clues about the function, the metabolic pathways, the signaling networks, and so on that are active in each cell in that tissue sample. And it's that resolution revolution that, to a large extent, was one of the drivers of the project dubbed the Human Cell Atlas. The vision of this project is to create a comprehensive reference map of the types and properties of all human cells as a basis for understanding, diagnosing, monitoring, treating health and disease. And it's a vision that Aviv and I put forward when we teamed up in late 2015, early 2016, as I was moving from the previous position where I was a group leader to the Wellcome Sanger Institute to head up a new department called the Cellular Genetics Department at the Wellcome Sanger Institute. And I reached out to Aviv Regev, who was in Boston at that time and was like-minded in terms of the possibilities that the resolution revolution in genomics was opening up to the community. And the shared vision that we had was to create a Google Maps of the human body using these genomic technologies, and to do it in a way that was truly international and community grassroots driven. And so the project now has over 2,000 members from over 1,000 research institutes from 76 countries across the world, and there are 16 from Austria. And we hope that there'll be more after this lecture. You can become a member of the consortium by simply registering at humancellatlas.org/join-HCA. And we welcome members from academia, from industry, from all the different disciplines that are relevant for tackling a challenge like mapping the cells in the human body using genomics technologies. And so our community is very interdisciplinary as well as very international. It includes clinicians, it includes biomedical experts, it includes technology developers and computational experts. What was important to Aviv and me was not only what we're doing in this project, but also how we embark on the journey of mapping the human body. And we wanted to have a very open community and a truly global community that embraces equity and diversity. And what we have fantastic partners in that, in terms of the co-chairs of the equity working group, who are shown here: Alex Shelik, Partha Majumdar, Musum Langa. And we are committed to carrying out the project in a way that includes a diversity of scientists and also a diversity of samples, so that we really are mapping the human body from all over the world. The way we've organized our community is in terms of not only computational and technological working groups, but also biological networks. And this really evolved during the pandemic, that we create groups that are coordinated by experts for the different systems. So we've kind of organized the body in terms of the major systems that you can see here, as well as organoids, which I'd be remiss not to mention here in Vienna. And this group is coordinated by Christoph Bock and others, whose, and of course the relevance is the comparison of these in vitro systems with the in vivo data from the Human Cell Atlas. And related to that is also the study of human embryonic and fetal development, as well as a working group on genetic diversity. The advances in technology, as I've already mentioned, are key motivating factors for this project. And it includes the single cell revolution, the resolution revolution in genomics in terms of suspension cells, but also the spatial genomics technologies that are developing extremely rapidly at the moment, and that allow us to put the individual cells, or to look at the individual cells in their native tissue context. And so really map cells in terms of the cake. And these technologies together are, of course, a world apart from the conventional bulk genomics, where we needed to have thousands or hundreds of thousand cells even in order to measure the DNA or the RNA of a given sample. And so this much more detailed access to genomic data, coupled with integration using computational methods, which are absolutely at the heart of the project. The computational biology community, is one of the first communities that rallied around the project, basically makes this project possible. And so it's these technologies coupled together with the computational methods that allow us to get from the coarse-grained view of tissues into the genomic, full genomic breadth description of single cells in their tissue context. And of course, that approach and that mapping of cells is leading to incredible discoveries in terms of our basics understanding of the cells, their tissue context, their signaling circuitry. But what's the impact of those discoveries in terms of the translation to the clinic. So Giulio very nicely mentioned that the objective of the CeMM is cutting-edge basic science research with translational impact. And really, that's very much how I see the Human Cell Atlas project as well. It's a basic science project about discovering and understanding our cells and tissues. But there are very clear connections to gaining a better understanding of disease mechanisms, which I'll allude to later in this talk, to drug discovery, understanding drug toxicities in terms of mapping where they act in the body, understanding epistatic interactions, diagnostics in terms of the technologies themselves being used as diagnostics, mapping of genetic variants, and of course, in terms of regenerative biology and understanding these in vitro systems, and I'll come to that later. So I'll try to give you vignettes that illustrate the utility of the Human Cell Atlas in terms of these two impacts, disease mechanisms and regenerative biology. And we lay this out in the Human Cell Atlas. And I should say that these impacts, or these different areas of impact, are likely one of the drivers for the huge enthusiasm in the community for this project and the huge enthusiasm of the funders for this approach. Examples of Human Cell Atlas data today for different organs and systems are shown here, just to give you a flavor of the amount of data that the community has generated together and how fast this project has advanced just over the short period that it's been going. And so for the kidney, for instance, there are 500,000 cells, including cells profiled from COVID patients. For the skin, many hundreds of thousands. The airways, which I'll come back to later, almost 3.5 million from over 300 individuals. So we're also sampling from many hundreds of individual donors. The gastrointestinal tract, almost 2 million across the small and large intestine. Human development, which obviously encompasses a lot of different tissues, almost 6 million cells, and the liver, 500,000. And so this is just to give you a flavor of the amount of data that's being accumulated by the community and the advances that we're making. I now want to shift gear and give you an example of an ongoing Human Cell Atlas project from my department at the Sanger Institute, from the Cellular Genetics department. And I want to credit Rosa Vento-Tormo, who was a postdoc in my group and then moved to a junior group leader position and is a rising star in science, basically in starting her own group, and has spearheaded this project together with her own group members over the past couple of years. And the reason that I want to present this particular work is that it's one of the motivations for the Human Cell Atlas project is understanding systems that are still poorly understood to date. And the human uterus is a good example of that, because it's an organ that has structures that are very specific to us humans. So in the uterus, we obviously have the menstrual cycle every month. So it's really not a single static organ, but it's a dynamic system that has at least 3 major phases: the menses, the proliferative phase, and the secretory phase, which is where the window of implantation of the embryo is, and the leading to the beginning of pregnancy. Sorry, I'll just change my cursor to laser pointer so that you can see where I'm pointing to better. And of course, the menstruation is something that's specific to humans and is not present in rodents generally. And therefore, mouse models don't give us the whole picture of what's happening in the human organ. And so that's one of the reasons why the human system is relatively poorly understood. The other reason is the dynamic nature of this process, so that you need a lot of different samples, and it's quite hard, obviously, to access these tissues from premenopausal women. And so in Cambridge, we've been very lucky in the sense that we have tissue access to full thickness biopsies of the uterus from our colleagues in medicine and transplantation, in the surgery, from the surgeons who carry out transplantations. And this is Koar Sayed Parsi and colleagues in the Cambridge Biorepository for Translational Medicine. And we have a close collaboration with them. And what they do is basically as tissues are taken for transplantation, very relevant to the Landsteiner Lecture, tissues are also taken for research purposes. And these can be quite large biopsies, up to 5 centimeters in size, which gives you the full thickness of the uterus through the endometrium and the myometrium, which is the muscular layer around the outside of the uterus. And that's very special because from biopsies that are taken for diagnostic purposes from living donors, the thickness of the biopsy is about 2 millimeters. And that really scratches the surface only of the endometrium and doesn't get through the full thickness. And so we're extremely fortunate to have access to both those different kinds of human tissue samples, both the full thickness biopsies from that give us the endometrium and the myometrium, as well as the endometrial biopsies that are taken for diagnostic purposes. And we can access these samples from donors that are in different phases of the menstrual cycle. And so for the deceased transplant donors, there are smaller numbers because of the small numbers of this type of donor that we can access every year. And so there are 4 donors in the follicular phase, 3 in the luteal phase, and these numbers are now somewhat larger since this slide was made, and then larger numbers for the small biopsies. And so in that way, we can start mapping the dynamic changes that happen using a combination of single cell and single nuclear RNA sequencing, which is the suspension single cell genomics technologies that I showed you, as well as spatial genomics using the 10x Genomics Visium tissue sections. And that's what I'm showing you here in these 3 different images on the top right and on the bottom. What these are thin sections of the tissue put onto a slide, and then the transcriptome, in other words, the RNA content of each of these little circular voxels is captured. And each voxel has about 20 to 100 cells. It's a 50 micron voxel. It's not quite at single cell resolution at this point in time, although these technologies are gaining in resolution and in the density of profiling. And I'll come back to how we can use this data to get down to a pseudo single cell resolution using computational methods. But here, what I'm showing you is the transcriptomic signature mapped onto these tissue sections for glandular epithelial cells on the one hand. And so wherever that signature is present that we get from the single cell data, there's a dark red spot, and this shows you that the glands start off here in the basal layer of the endometrium. This is the myometrium down here. And as you get to the top of the slide, it's the basal layer of the endometrium up to the luminal layer, which is the inside of the uterus. And you can see the glandular epithelial cells that do those secretions that support the embryo in these dark red spots along the endometrial layer. What we can also map is different types of fibroblasts. Now, if the epithelial cells are sort of the business people of the tissue who carry out the key functions of each tissue in the inside of our body, the fibroblasts are sort of the infrastructure elements that provide a supporting architecture in the tissue. And in the endometrium, that's particularly true in the sense that the fibroblasts are probably as abundant in the tissue as the epithelial cells that I'm calling the sort of business end of the tissue. Fibroblasts are structural cells that are also important in wound healing. They support the tissue, and they provide different microenvironments in the tissue that I'll explain later. So here, this is showing the expression signature for a particular type of fibroblast, decidualized stromal cells, we're calling them, and they sit on the top of the top layer of the endometrium, close to the lumen. And these fibroblasts are really important for supporting pregnancy and contributing to the tolerogenic immune environment when the embryo implants and the maternal immune system is presented with paternal antigens, which is a kind of conundrum that's akin to a transplantation situation where the maternal body is presented with antigens that are foreign to it. And of course, in this situation, the amazing kind of magic of the uterus is that it supports those foreign antigens and doesn't reject them as in a transplantation. So I'm sure Landsteiner would have been very interested in this system. in solving this puzzle, how the immune system can actually tolerate these foreign antigens. And that was one of the things that made me so curious about this tissue, because one of my central interests over the past 10 years or so has been T cells in the immune system. And in other types of fibroblasts are shown here. We have a particular type of fibroblast that's in the basal layer and that's different from those decidual stromal cells. And you can see their location is down here at the entry point of the endometrium to the myometrium. So we have those infrastructural cells defining different layers of the endometrium, which basically tells us in a very quantitative and molecular way that the zonated architecture of this tissue is defined by these very specific cell populations of the fibroblast compartment. Now, if we go back to the epithelial cells and go back to that business end of the function of the tissue, then we have ciliated epithelial cells here, and they have a very specific localization, again in the lumen on the inside of the uterus. Interestingly, we also have a progenitor population, so a kind of adult stem cell-like population that's also located in that top layer that's shed every month. And these LGR5 positive progenitors are one of the cell states that we discovered in the course of this project. Now, how do we map those cells to their particular locations in this coarse-grained transcriptomic map? We used a cell-to-location algorithm developed by another rising star in my department, Omar Bayraktar, with Oliver Stegle and their group members. And this cell-to-location algorithm takes the single-cell data and maps it to a spatial location in the tissue based on that coarse-grained spatial transcriptomics data that I just showed you. This is an example of a lymph node and the single-cell data for the different cell types here, and they're placed in their positions in the germinal centers and the different zones of the lymph node using that cell-to-location algorithm. So just using this movie, what I hope to transmit to you is how important computational methods are for the Human Cell Atlas project and how key it is for us to be able to develop these probabilistic models, machine learning models that integrate different data types with each other and create the three-dimensional map of the Human Cell Atlas. Now, of course, we don't forget microscopy. We still rely on microscopy as a way of seeing individual molecules at cellular, subcellular resolution. And for the endometrium here, we have staining of four different markers simultaneously that mark different epithelial cell populations during the different phases of the menstrual cycle. So these are samples from proliferative, mid to late proliferative, all the way through to the late secretory phase, that implantation phase at the end of the menstrual cycle. And you can see here we're zooming in at higher and higher resolution, and we can actually see how the glands are forming and how they form a plumper structure basically throughout the late proliferative phase when the tissue is building up after the menstruation, and how the individual epithelial populations migrate and locate within the secretory glands. And at the lumen here, this matrix metalloprotease positive epithelial population is now luminal here on the top of the endometrial surface at the end of the menstrual cycle. And it's this sort of high resolution microscopy information that basically validates, is really helpful in validating some of the discoveries and telling us about detailed morphology of cells and microanatomical structures. And this is work by Kenny Roberts with Omar Bayraktar, whom I already mentioned in the cell-to-location algorithm. And so amongst other cell states that we discovered in this work, we discovered three different progenitor populations. I've mentioned that LGR5 positive population that we position in the top, in the luminal layer, as well as proliferating progenitors, and a SOX9 progenitor that's further down the basal layer. And so this is really, I want to emphasize that these progenitors, have been described for the first time in human. They're really different from our rodent furry friends, or the mouse counterpart that's used so often in the lab. And it sort of illustrates the value of studying human biology in terms of the reproductive system. And there are many similar discoveries in the immune system, which is also a fast evolving system. So Giulio mentioned that I have an interest in evolutionary biology, and certainly different systems in the human body are diverging from our primate and rodent cousins, if you will, at different rates. And reproductive biology, immunology are evolving very quickly because there's different types of selective pressure on them, whereas other systems are certainly evolving at slower rates and are more similar to our animal counterparts. Anyway, so the interesting thing about these progenitor populations is that they can inform us about the identity of endometrial adenocarcinomas, and the LGR5 positive population, that particular progenitor cell, is enriched in serous endometrial adenocarcinoma, whereas the SOX9 positive LGR5 negative progenitor is more enriched in the more aggressive endometrial adenocarcinoma. And so the discovery of these different progenitor populations allows us to interpret cancer data in new ways and understand these tumors in new ways that can give us information about how to better diagnose them and how to better treat them going forwards. That's the hope. And so in that way, the human cell atlas data can be really informative with respect to diseases, and the comparison of healthy reference data allows us to understand and interpret tumors in much more detail. And the same is true for many other tumors, including pediatric tumors. If we go back to the basic biology of the endometrium, what we can do here is use Dorothea, which is a gene regulatory network inference framework from Julio Saez Rodriguez, and predict which transcription factors are key for which cell lineage based on expression of downstream expression modules. And so for the ciliated lineage, which are those cells on the inside of the uterus that I showed, we find two key transcription factors that are linked to Wnt signaling, FOXJ1 and HES6. So these are two specific factors that are linked to the receiving of signals from a specific signaling pathway called Wnt. If we look at the glandular epithelial cells, which are those that do the secretions that support the embryo, we find other transcription factors, HES4 and HEY, that are implicated in signaling from cells down the notch pathway. So there are two different pathways for these two different lineages of epithelial cells, which are those cells that, as I mentioned, sort of carry out the business of the uterus in terms of the secretions for the embryo and the forming the lining of the uterus. So again, if we go back to those kind of infrastructural cell populations that I told you about before, which are called fibroblasts, then we can position them in the different regions of the uterus. Here, I showed you the basal populations, which are in red and orange down here, the so-called functional populations, which are in the middle of this zonated tissue. So these infrastructural cells really form that zonated architecture of this tissue that goes from the myometrium into the lumen of the uterus, the luminal populations. And we can now really sort of see this, not just in a morphological sense under the microscope, but we can map this using genomics for the first time and actually unravel the functions of these cells. And in particular, what we can unravel is what these different infrastructural cells are, what signals are they sending, and which of these are sending Wnt signals and which are sending Notch signals. And it turns out that in that functional layer where the glands are located, there's Notch2 being secreted, and we can validate that by, again, with microscopy, by staining for the Notch2 signal. And so these humble fibroblasts that are forming these structural layers are not only kind of passengers, but they're actively secreting signals that are instructing the epithelial cells, which are the kind of functional business cells of the cell, and telling them what identity they should take on. And from a computational point of view, the way we can systematically make these inferences and organize this data is using our database and statistical framework called CellPhone. So this is a database that encodes all the cell surface ligands and cell surface receptors which cells use to talk to each other. So this is the cellular communication network, if you will. And what CellPhone does is encode all the different genes that are necessary to make these complexes, and then provides a statistical framework for inferring when cells are expressing complementary sets of these receptors and ligands. And what we also take into account in the new version of CellPhone is where they're spatially coexisting, where they're in the same spatial microenvironment. And this allows us to predict high confidence cell-cell interactions that are mediated by specific complexes. And so I've told you that Notch is secreted by these functional fibroblasts that are signaling to the glandular epithelial cells and telling them to differentiate down that lineage, and Wnt to the ciliated epithelial cells that are at the top in the lumen. And so that allows us to really make a sort of model for the way this tissue is structured or organized between the fibroblasts and the epithelial lineages that involve signaling using Notch from the fibroblasts that are near the glands and that encourage the progenitor to make glandular epithelial cells, and Wnt for the fibroblasts that are in the lumen that are encouraging the progenitors to make ciliated epithelial cells. And these are the particular signaling factors and the particular cell intrinsic transcription factors or regulatory factors that are controlling this cellular decision-making process. So obviously, having specified all these signals that are coming from the outside of the cells and the signals that are on the inside of the cells sort of gives us the recipe for engineering these cells in the dish. And so I want to put forward this idea that the Human Cell Atlas essentially gives us the instruction manual for making cells and organoids in vitro. So you can think of, for the endometrium, that the Human Cell Atlas of the uterus is telling us which cell surface receptors and which ligands are signaling to each other, what we need to put into the medium, and which transcription factors are therefore being switched on to make the secretory epithelial cells and the ciliated epithelial cells in the endometrium. Now that's our prediction. Can we actually make it happen? Well, we teamed up with Margareta Turco down in the pathology department at the university in Cambridge, and she developed an endometrium in a dish model system a few years ago. And what we predict is that we can improve that system by engineering it through the addition of Wnt and Notch inhibitors. So through the manipulation of those signals that I just mentioned are coming from the fibroblasts, from the infrastructural cells in this tissue. And we can then characterize those new organoids, both by microscopy in terms of the shape and morphology of the cells, of the new cells, but also by single cell RNA sequencing itself, and basically have a quantitative comparison of whether those new organoids are more faithful models of the epithelial cells in the tissue in vivo. And this sort of assessment or quality control of organoid systems through quantitative comparison with the in vivo Human Cell Atlas data is one of the visions of the organoids working group of the Human Cell Atlas that's co-chaired by Christoph, whom I mentioned earlier. And I hope that sort of illustrates also how useful the in vivo Human Cell Atlas data can be, because it really allows us to carry out precision engineering, if you will, in the dish. And obviously that, as I'm sure you're all aware, has huge utility for drug screening, ultimately for actually engineering tissues that can be transplanted for regenerative medicine purposes, or for cells that can be used for cell-based therapies as products. So back to the endometrium, these are little icons of the different cell states that we discovered in the course of this project. The progenitor cells are shown up here that are particularly novel and interesting. This SOX9 cell in the bottom, in the basal layer of the glands, a proliferative intermediate, and then a sort of transit amplifying cell, and then a different progenitor that's more in the luminal layer, and these are the differentiated, ciliated, and glandular cells. And in gray here, you can see the different infrastructural layers in the endometrium that are formed by those fibroblast populations that I showed you, which play a passive or a structural role, but they're not passive, they're actually instructive, as we show here. I've mentioned Rosa, and I've mentioned her group members, and there are many others that contributed. This was a real team effort, including Omar Bayraktar's lab, Christoph Polansky from my lab, support teams from the Sanger Institute, and our collaborators down in the university, Margarita Turco and Ashley Moffat and their group members, also with the Protein Atlas, and our transplant surgery colleagues, Krishna and Korosh, and the biorepository for endometrial biopsies at Newcastle University and Paul Ayer. I want to hope that that's illustrated the use of the Human Cell Atlas for regenerative biology. I want to give you a very quick last whistle-stop tour of insight into disease mechanisms before I finish. One of the early Human Cell Atlas projects from my group was mapping the airways. And the view that we were taking here, studying both the upper airways, the nasal passages, the bronchi, and the alveoli, was basically a mapping of the cells in those tissues, but also a comparison to biopsies from asthmatic donors. And so that was really the perspective that we were taking in that work. And this is a collaboration with colleagues in the Netherlands, where they took bronchial biopsies from healthy and asthmatic donors. They also took nasal biopsies to kind of profile the inside layer of the nose. And we profiled alveolar tissue from deceased transplant donors. And we used suspension cell, single cell genomics to map the different cell types. And what we noticed in the nasal passages was that there were two particularly interesting goblet cell populations. So goblet cells are, again, epithelial cells, those business people that are carrying out the functions of the tissue. And they actually have certain similarities in a distant sort of sense to the secretory cells in the endometrium, those glandular secretory cells that I was just talking about just now. And these goblet cells were particularly exciting to me because this population had an active cell circuitry that we predicted to be talking to T cells and dendritic cells as they come out of the vessels into the tissue. And then when the pandemic hit, we went back to our airway data and found that precisely that cell population that had this active immune communication program had very high expression or particularly high expression of ACE2 and the other viral entry receptors for the SARS-CoV-2 virus. And this basically then led to a cascade of collaborations that allowed us to map the viral entry receptors for COVID-19 across the whole Human Cell Atlas data that the community had assembled during that time. And the really exciting and encouraging thing, in the sort of tragedy that was the pandemic in those early days, back in February and March of 2020, was how the Human Cell Atlas community came together and shared unpublished data basically overnight. It was really, really amazing. And it was the Human Cell Atlas lung biological network that was formed throughout this public health emergency, and that coordinated, that helped to coordinate this effort. And so this is work that was spearheaded by a postdoc in my group, Bordon Songnak, with a staff scientist called Ni Huang, and with the support of the coordinators of the lung biological network and the collaboration of the whole Human Cell Atlas community that allowed us to map the viral entry receptors for SARS-CoV-2. And we also compared to influenza and MERS across all the tissues, or at least 26 different tissues of the human body. So there are 50 in total, but at this point we had data for 26, so just over half of the tissues. And I'm going to focus just briefly on the tissues that are at our interface with the outside world, and that includes the airways from the nasal passages down to the alveoli, the eyes, the gut, and the uterus, where the interface with the fetus. So in the upper airways, I've mentioned that secretory goblet cell population that had high levels of receptor. And that's incredibly important in understanding the transmissivity of the virus. When you sneeze, for instance, it can jump very quickly from one person to the next because the virus is sitting inside those cells. And we now have evidence for that in the meantime from direct evidence. In the lower airways, in the bronchi, they are both club and ciliated cells where the virus can enter and sit. And then, of course, deep down in the lung, it sits inside the alveolar type 2 cells. And that's, of course, where a lot of the damage occurs in severe COVID-19 patients. And so the healthy reference human cell atlas data assembled very rapidly and analyzed in this way is giving us insight into transmissivity and also into sort of later pathology of the virus. And in the eye, this was data that Linda Lako shared just as her lab was shutting down. And she just threw the data over to us, pushed it over to us, and we found these superficial conjunctival cells that had high expression of ACE2 and TMPRSS2. So these are cells that are in the whites of your eye, and this implicates these cells in terms of understanding the viral conjunctivitis that's been observed in COVID-19, and also in terms of implicating the nasolacrimal tract in transmission across individuals and within the body. In the gut, enterocytes in the small and large intestine have high expression of these receptors, explaining some of the diarrhea potentially that you see, particularly in pediatric patients. And at that maternal fetal interface, which we studied in detail in first trimester pregnancy a few years ago, we find perivascular cells and those decidual stromal cells that I presented to you, those fibroblasts in the top layer of the endometrium and the pregnant state of the uterus, they actually have the viral entry receptors as well as trophoblasts on the fetal side. And so this gives a potential mechanism for maternal fetal transmission, which is obviously rare, but it does happen. And then we also map many other tissues in the body. And I've acknowledged the two superstars who worked incredibly hard over a short period of a few weeks to assemble this, and the entire human cell atlas community that came together as an incredible network to make this happen. And we served this data on a website that's been accessed tens of thousands of times. This work has been cited thousands of times, or 12,200 times roughly. And we hope has contributed also to the policy making around masks in those early days for covering the nasal passages. Now, the Cell Atlas community has gone on to study the blood in different cohorts of patients, tissue samples, and organs from autopsies from deceased patients. And we again serve this data online here in terms of not just the healthy reference data that I showed, but also patient data. And in the in nasal scrapings of patients, where we're actually getting nasal samples from patients, we can see that yes, indeed, those goblet cells that I predicted have the high ACE2 and TMPRSS2. We show that they actually also harbor viral reads, and so we can actually sort of see the virus using single cell genomics in these individual cells, and see that the cells that we predicted have the high viral receptor levels also do actually harbor the virus inside them. And the same is true in the oral cavity, and this is a collaboration with colleagues at the NIH and University of North Carolina, Blake Warner and Kevin Bird. And again, Ni is the one who analyzed this data. And we show that in the from biopsies from human, the human oral cavity as well as the mouse, there are cells in the salivary glands, in the specifically ductal epithelial cells in the ducts of the salivary glands that have high TMPRSS2 and ACE2 levels. These are shown in the red dots and the gray here. You can see it inside the ducts of the gland. And in the infected patients, COVID-19 patients, we can see the actual SARS-CoV-2 viral RNA in exactly those cells. And so this imaging data basically, again, validates our prediction that the cells that have the high viral entry receptors in these salivary glands are also the ones that are welcoming the virus. And of course, this is incredibly important, again, in terms of understanding why when those cells are shed in saliva, this virus is so highly transmissible. The reason is that it's sitting inside our salivary glands. Those cells are shed into the saliva, and the virus can actually jump from one person to the next in that way. And so in that vein, I hope I've convinced you that the human cell atlas data has a utility for medicine in terms of a detailed understanding of disease mechanisms and the public health implications of that. And in the earlier example of the endometrium and the organoids, regenerative biology. And I've told you about human cell atlas work on the airways and defining the different cell states in the airways and also in the female reproductive system and developmental biology. Thank you for listening. It was an honor and a pleasure, and I'd be delighted to take questions.
Thank you very much, Sarah. This was indeed a fantastic talk, very worthy of the Landsteiner Lecture, and you cited Karl Landsteiner, who in his grave or in his paradise will be very pleased to get his acknowledgement. So there are questions in the chat. I would like to start with a fundamental question. I will ask the question in the chat, don't worry. A fundamental question is an obvious question somehow, which is you're looking at all of these different cell types and states. Is there a sheer infinite number of such cell types and states because it is a matter of philosophy to define what differs a cell from another? Or do you think there is a finite number so that you can say these are all permutation of states around one particular cell type? In other words, when I was a student, we thought that there would be 200 cell types in the human body. What can the lay audience listening in estimate in terms of different types of cells in the human body?
So it's a great question, Julian, one that I get asked a lot. And it would be, at this point in time of the Human Cell Atlas project, it would be a kind of lottery for me to give you a number of cell types. I mean, I mentioned to your Viennese journalist colleague who interviewed me for the radio earlier that the number. I think it's safe to say that we're talking about thousands of cell types, not hundreds in the human body. I think that's pretty safe to say. But you're absolutely right that you can kind of refine that definition into many different metabolic states, cell cycle states, stress states, developmental states, and so on and so forth. And so if you really want to split hairs in that way, you could kind of up that number, again, by 10 or 100-fold. But I think what we'll probably converge on is using words to describe the major cell states. Now, bear in mind that, speaking as a computational biologist and from a computational point of view, you can describe things computationally and the trajectories going from progenitors to the differentiated cell states without needing words, really. But I think it's very useful and very helpful concept to describe individual cell states as the endometrial progenitor, let's say the endometrial stem cell. It's incredibly powerful and helpful to us human beings to have those words. And so what we'll probably end up with ultimately is a set of names and words for cell types that capture the core, these core kind of states and their functions.
It's going to be a cell ontology that you will probably have to drive to, and so before we move into more, let's say, philosophical questions, there are a few questions from the audience that have to do with understanding what you have presented. The first question addressed whether there is a reason why you left out the mark, sort of the blood mark, the Knochenmark.
No, there was no, that was, no, the bone marrow is absolutely fascinating as the place where we make hematopoietic stem cells in our body. And, we'll soon release a paper where we studied the actual development of the bone marrow. And I think it's fascinating and it's key tissue for the human cell. It was absolutely, yeah. So I was just trying to cover a few. I
was reassuring the person who asked the question, who is a person that has a lot of initiative on specific rare disorders that affect the hematopoietic system, to say this, and I anticipated that you had not forgotten it. Then there is another question on understanding. Andreas Berg would like to understand what is the spatial encoding that you're showing. How do you go from, if you want, a transcript analysis to being able to say that cell was there and there and there? And he proposed several ways by which you're doing it. I don't think that people quite understand that.
Yeah, so there are two major branches of spatial technologies that are whole transcriptome or very, very highly multiplex. One is the sequencing base one, and that's what I was showing. And that's where you have a chip that has barcodes on the chip. And when you put the tissue section on and you lyse the cells, the RNA is released and captured by the barcodes that are on the chip. So that the spatial encoding is achieved by the barcodes and the position of the barcodes that are on the chip that are defined a priori by sequencing. The second category is imaging based, and those technologies rely on probe sets that are read out by combinatorial fluorescent markers, and so they're generally not fully transcriptome wide, but they're very highly multiplex. And so that's the in situ sequencing, Murfish, Seqfish kind of technologies belong to that category. Now, a few days ago, 10x Genomics, I think it was either yesterday or maybe today or yesterday, described a third kind of category that basically integrates the strength of the two, and which works on FFP tissue, where you have probes. So it's like the imaging-based branch that I mentioned, and there are probe pairs for every single transcript in human or mouse genome. But those probe combinations are then read out by next generation sequencing. So in a way, that's a third category. I hope that answers your question, Andreas.
Thank you. I think it did exhaustively. There is a question, there are two questions by Federica Quattrone, one of our students, but I think the second one is probably one that people are curious about, which is, she says that in the past, people have proposed that at implantation embryo, there is a quality control that occurs over the endometrium. She asked whether you see any, whether you can see any evidence of that, maybe by the kind of things that are expressed in the progenitor cells or in the other cells there.
So for the lesions of patients affected by endometriosis, I mean, that's one of the things that Rosa Ventotormo and is actively working on together with Krina Zondervan in Oxford. And I think it would be really fascinating to compare the endometriosis lesions with the healthy endometrium, and because it's such a devastating disease, there, you know, very few therapies, incompletely understood, and that's ongoing work. And that I didn't mention endometriosis, but it's one of the major applications of the endometrial cell atlas. Whether the endometrium plays a role in quality control, I find it hard to comment on that. I mean, there are human genetics indications. I'm not sure if you're aware of the work from Ashley Moffat, Peter Parham, and others saying that there are some HLA KIR incompatibilities, basically between maternal KIR and paternal HLA, and they explain about 13% of cases of preeclampsia. And so that's a kind of human genetics epistatic interaction where there's a sort of a lack of tolerance basically for the paternal HLA in the maternal KIR cells. But that's sort of different from a quality control. I mean, yeah, so it's an interesting question that you raised, Federica, and one that I can't completely answer.
So there's also a question that concerns the use of matrigel. You know, is there a non-cell origin source that you would consider as the matrigel that we use typically is produced by mouse sarcoma cells? Ask Philipp Schwabel.
Well, I think the support is it's an interesting question, and I certainly agree that the support doesn't accurately mimic the human organ support structures for sure. There are alternatives in terms of collagen, human collagens that are added to form a solid support structure. There's much stiffer material from a material science point of view, and I think all of these sort of progressions in making better in vitro systems will contribute to making more faithful models of the human tissues and organs.
Thank you. There is a question that speaks about the aging of the immune system in children. So if you see during development, if you see changes that would speak to...
I think that's a fantastic question, really fascinating, because as you may or may not know, the thymus is one of my favorite organs. And of course, the thymus develops during essentially the fetal stages of pregnancy, so the second trimester, third trimester, and is kind of at its best right after birth. And then when we hit puberty, the thymus starts involuting. And the reason that it's so important is that it's the site where our T cells are made and where they're trained to distinguish self from non-self. So it's that essential component of the immune system that poses such a problem for transplantation. And it's that conundrum and mystery of how the fetus is tolerated at the maternal fetal interface. So the thymic aging, I think, is one of the reasons also the kids clear SARS-CoV-2 so quickly and effectively. And we have, so the Human Cell Atlas, I don't know if it became clear from my talk, but it includes, we include not only the adult Human Cell Atlas, but also embryonic, fetal, postnatal, and so on into the project. And I do agree that we can really rewrite textbook knowledge about aging of the immune system by studying postnatal stages. And we have that paper that I showed, and maybe it went very quickly, but the nasal swab data where I mentioned the goblet cells are infecting the virus, that's online on medRxiv, and we profile actually large numbers of pediatric healthy donors and pediatric COVID patients. And we show that the T cell output and the T cell clonalities are completely different in children compared to adults. They have a much richer oligoclonal repertoire and response to the virus because their thymic output is so active and rich. And so kids are really, it's the first time seeing is believing. It was kind of known, of course, that the thymic output is different in children than adults, but it's so clear in that data. What's also interesting is that the transcriptomic identity of the naive T cells in CD4 and CD8 compartments is much earlier, kind of more thymic-like in the kids than it is in the adult patients. I think it's a really exciting. Thank you for that question.
So that was from Kan Bostuk. And Christoph Bock is asking also a question that is fairly complex about the evolution aspect of the uterus. Can you see a molecular basis for pregnancy being developed in evolution? Can you see something special about the fact that you have this? And my question that goes with it is it now very interesting to go and do some primates and do other animals?
Exactly. So that was going to be my answer, Giulio and Christoph. So I think it will be fascinating to do the same thing in our non-human primate relatives. And, I know that there are colleagues in Heidelberg, Duncan Odom and collaborators who are studying many different rodents in terms of their diestrous cycle. So it's not a menstrual cycle, it's an estrous cycle. And I think that will give us really interesting insights into the basis of mammalian pregnancy and evolution. And ultimately, it would be fantastic not only to have a human cell atlas, but to have a cell atlas of lots of animals. And then we'd understand the evolution of cell types and processes like this across different parts of the body and different parts of the animal tree of life.
There is a question in the panel that says, do you see a future where single-cell RNA sequencing could be used for diagnostic or even screening?
Absolutely. So, just the way the Human Genome Project led to, whole genome sequencing for cancers and for rare disease, it's now used routinely in the UK National Health Service for those specific disease categories because whole genome sequencing has become so cheap and accessible. And the bioinformatics is mature. I think in exactly the same way, single-cell RNA sequencing will become a diagnostics and screening technology maybe 10, 20 years from now, and spatial transcriptomics will become a pathology technology, essentially, next-generation pathology.
So now I would like to ask you a few more general questions. One is, if you could do magic, or in a few years it will be things that will come out of your laboratory, would you opt for being able to have at the single cell resolution, transcript analysis, epigenomic analysis, metabolomic analysis, protein interaction analysis. If you were to choose one thing that condenses the knowledge and it is in future, I think, most information dense, what would you think it could be?
I mean, I'm sure, as you're aware, Giulio, there are now multi-omics technologies that allow you to measure multiple modalities from the same single cell. And that includes, RNA and epigenome, it includes RNA and protein, and so on. And so we won't have to choose. My ideal wish would be that we can have everything from the same single cell. But if I was really, really, really forced to choose, my feeling is that the epigenome is kind of the most direct interpretation of the genome. And that it goes beyond coding regions, of course, as you know, into all the non-coding regions of open chromatin and histone modification. So if I were forced to choose one, I might choose that one as a sort of data junkie.
Thank you. Presumably one of the last questions still from me is to say, studying your CV, it's clear that there is a phase where you are cataloging things, and then there is a phase where you're moving to the laws that govern the organization of those things. So as now you're seeing many, many tissues, and you see the different state of development, and you see the different organization of how tissues arise and acquire function, what could be a meta-level analysis where you try to see every time there is this kind of cell in any tissue, there's also that kind of cell, and do that really computationally, so not just anatomically, but based on data?
Well, I think there will be patterns of essentially, combinations of cells that emerge once we integrate the human cells of many different tissues. And it may not be always the same types of cells, but it could even be a sort of meta-analysis or meta-level where there are particular signaling pathways that are going from one cell to the next. They may be occupied by different cells. So that's when we compare human to mouse data, we sometimes see that the same complementary signals are sent by different cells in the mouse as compared to the human. So although the signaling is there, it's not carried out in quite the same way. So there's been kind of tinkering in evolution. I think that, essentially the distilling down the function of a tissue and of cells to their gene expression and signaling programs is essentially doing what you're saying, kind of.
The periodic table of tissues. I mean, what is it? So I would like to finish with two questions that come from two CeMM students. I read them out because they are complex. One is by Sasha Petrenko. He asked, you see it in your chat, ligand-target interaction is a fascinating topic. And during the talk, you discussed the application of the cell phone DB database, which is based on ligand receptor expression level in the data set. What do you think about models additionally relying on perturbation experiments and signaling networks, such as NiCakeNet? Could they be more beneficial for studying mechanism in healthy versus disease? And I think we can also take the second question together, which is, would you like to imagine that you were able to study aging using your analysis?
Sure, thanks for those questions. So in response to Sasha's question, I think the perturbation studies are fantastic, and NicheNet is a brilliant tool. Cell phone, would greatly benefit from I think systematic perturbation experiments. What we were trying to do with cell phone in the first instance is to create a resource that takes into account the complete stoichiometry and the requirement for all the different subunits of a protein and of a protein complex to be present. And to my knowledge, it's the only one of all the different resources that does that. And it sort of relates to my background in protein complex assembly and the structure of protein complexes that Giulia was mentioning earlier. The second question is whether there are plans to study aging, and the answer is absolutely yes. I mean, there are groups within the human cells community who are really interested in aging, and including my own group, and Steve Quake has released sort of several aging projects and data sets recently, which are incredibly interesting. I would say that's a sort of next generation human cell atlas project because of the simple cost of studying human aging, because it's so variable. There's differences between male and female. There's differences in every decade of life. So it's really a very sort of expensive human cell atlas project if you want to do it properly. But it's fascinating, and I think it will sort of be the next wave of human cell atlas after kind of version 2.0 is complete, let's say.
Thank you very much, Sarah. I think we finish here. I mean, I think that everybody who witnessed this lecture will remember it, because these are days where you're reporting on exploring tissues that is in every one of us at one stage, or most of the tissues, maybe the endometrium not in all of us, but sort of different tissues, and still you're discovering, you're newly discovering, and you're a member of the Royal Society. To me, it's like a member of the Royal Society in the 19th century coming back from some exploration and reporting on new kind of beings and places and plants and so on. So it is exploration date. You're a fantastic, intrepid explorer. We are privileged to have gotten a prime report from the unknown, and we wish you success in the continuing, I don't know how to speak tonight, continuing sort of adventures that you're taking. Thank you very much. Come and visit us. And the next time, the lecture will be in the Festival of the Academy of Sciences. So thank you very much. If anybody wants to join with an applause, turn on your camera. Maybe we can do that, and you can all join us in some Zoom applause that is more than deserved. Thank you. And thanks to the prize winners. Good night for those of you who are in this part of the world.